to introduce today's seminar speaker who's helping us kick off this year's seminar series. And it's actually a really big treat to get to introduce my colleague, Dr. Laura DeNardo. Super quick bio, so I don't take too much more of her time. Laura did her Ph.D. at UCSD, where she made some really key contributions to our understanding of molecular mechanisms that underlie development and functioning of hippocampal synapses. She did her postdoc at Stanford, where she developed both viral and transgenic tools to help us map and understand the functioning of cortical circuits. And in particular, her TRAP2 mice, which have been broadly used throughout the global neuroscience community at this point, allow genetic access to specific circuits to better manipulate them and understand how they're really informing behavior. Laura started her lab here at UCLA in January of 2019. And I can attest to this firsthand because our labs are neighboring. She rapidly built a really vibrant research team. And over the last 6 years, that team has been incredibly productive and made a lot of really exciting contributions to our understanding of how circuits regulate behavior. So please join me in welcoming Laura, and we're looking forward to hearing about the stuff from your group.
All right, thank you. Thank you so much, Lindsay. That was a really nice intro. And so like Lindsay said, my lab is interested in how neural circuits drive behavior. And we're interested in how these circuits develop and how they are impacted by things like early life adversity. So today I'm going to talk about 2 stories that have to do with long-term memory, one in the developing brain, one in the adult brain. I like to start with this little anecdote about long-term memory. So these are my 2 kids, Kaya and Noah, and this is a picture from when we went to Palm Springs for Kaya's 6th birthday. So Noah was 2, Kaya was 6, and this was a little over 4 years ago. And they did all the same things. They saw the famous windmills outside the car window. They swam in the pool. They played in the sand. They ate pancakes that were shaped like unicorns that their grandpa made. And they both slept in their astronaut sleeping bags. But if you talk to them about this today, Kaya has a very clear memory of that trip to Palm Springs. She remembers this beautiful desert and how much fun we had. And Noah has no memory of this trip because he was only 2 when we went on it. So we're really interested in, why did Noah forget this? Why don't we remember experiences from when we're very young? And also, when we're older, how does the brain encode and then consolidate memories for long-term storage? So my talk's going to have 2 parts. Why can't we remember experiences from when we're very young? And then how does the adult brain store memories across time? And so I like to start with a little bit of the history of what we know about the mechanisms of long-term or remote memory, which is really mostly from studies of adults. So this is an experimental psychologist, Ribot, who in the late 1800s reported on this pattern he had noticed in a lot of patients he had. So people were coming to his office that had experienced some sort of trauma, and he noticed that they often didn't remember that traumatic event or the events leading up to it, but their memories of more remote events were intact. So this was some of the first descriptions that older memories are less prone to disruption. And in the 1950s, from people like patient H.M. and others who were undergoing experimental surgeries for a variety of reasons, we started to understand a little bit more of the brain mechanisms that are important for either recent or remote memory. So patient H.M. had a really severe case of epilepsy, and he decided to go and have an experimental surgery to try to reduce his seizure frequency. And what they did was remove his hippocampus bilaterally. And while this did help his epilepsy, the effects on his memory were really catastrophic. So Brenda Milner was a psychologist who studied patient HM for the rest of his life. And here's just an excerpt from one of her papers about what happened to him after he recovered from surgery. This young man could no longer recognize the hospital staff or find his way to the bathroom, and he seemed to recall nothing of the day-to-day events of his hospital life. There was also a partial retrograde amnesia, and that he didn't remember the death of a favorite uncle 3 years previously. but he could remember some events just before his admissions, and importantly, his early memories were vivid and intact. So this started to point to this idea that the hippocampus is important for encoding and formation of sort of short-term memories, but more remote memories must be stored somewhere else, because you don't necessarily need your hippocampus intact to recall those memories. And relevant to my talk, most of his cortex was still intact here. So these types of studies led to the systems consolidation hypothesis, which has then further been supported by a lot of rodent work, which says that initially these episodic memories are encoded in the hippocampus, which is highly interconnected with the cortex. And then through activation of these connections, a cortical network strengthens. And so that in many cases, you don't necessarily need your hippocampus to recall an older memory. And so my lab is really interested in what's happening in the cortex. So this is obviously a very simplified cartoon, but we know some of the key regions that are important for remote memory, but we don't know as much about which cell types are important with those regions. And of particular interest to my lab is which of these connections are really important, what's strengthening, like what does this network look like and how does it form? So today I'm going to kind of focus on this cortical network. And in this first part, we're going to try to understand how these cortical networks change across development and whether any of those changes may contribute to this infantile amnesia. And then in the second part, we're going to talk about a study that's looking at which cortical connections are particularly important for consolidating and retrieving long-lasting memories. So we'll start here. So we do know some of the mechanisms that contribute to infantile amnesia, but most studies have focused on the hippocampus. And what they've shown is that these early formed memories are not erased. They seem to be stored in the brain in a latent state so they become inaccessible, but there's still a trace of them in the brain. So some of the evidence for this comes from Christina Alberini and Paul Franklin's labs. So for instance, a lot of them use fear conditioning as a model, and so does my lab. And so you can fear condition an infant mouse, and you can test it later and show that it's forgotten that memory. But if you give it a very strong reminder shock, which is not enough to form a new memory, you can bring that memory back. So that was one piece of evidence that there's like a latent trace of these memories in the brain. Paul Franklin's lab showed that if you tag neurons that are active when an infant forms a memory, then they forget. You can reactivate those hippocampal neurons and also bring that memory back. So these are some of the key pieces of evidence that show that these infant memories are forgotten, but there's still a trace in the brain. And they also showed that the infant forgetting is associated with things like naturally elevated hippocampal neurogenesis, that rate declines as we grow up, and also immature hippocampal synapses and plasticity. So these are really compelling studies. And my lab is really interested to follow up on this work and try to understand why if there's a trace of this memory in the brain, why does it become inaccessible? And so we think that looking at the cortex is really important as a region that's, we know the hippocampus is initially involved. So something must be happening to these memories during the process of consolidation, but we actually know very little about how these memory consolidation circuits mature and how do their functions change across development. So today I'm going to talk about a study that's led by Benita Jin, who recently graduated from the lab and is now doing a postdoc at Columbia. And she's been working together with Michael, Abby, and Brian. Some of them are right there in the audience. And they're using contextual fear conditioning to model infantile amnesia in mice. So in these experiments, what we do is we pair a previously neutral context with a foot shock. And so these animals form a negative association with this context. And what they did was they trained animals these are mice, either at an infant age, postnatal day 17, a juvenile age, postnatal day 25, or early adulthood, P60. And then we wanted to make sure this model was working in our hands. So Benita just trained the mice and tested one group 1 day later and a second group a week later. And so you can see that at 1 day, infants, juveniles, and adults all freeze. So even the infants initially encode and retain a memory, but by a week that infant memory is gone. They don't freeze anymore, whereas both of the older age groups do. So we decided to go ahead and use this model. And then Benita wanted to start by sort of screening the brain. Like, these cortical networks that are involved in long-term memory are distributed. And so we didn't know exactly where to start. So she said, let's do a screen to look for where the activated cells are in these different age groups and see if that will point us to a good region to look at first. And so they used these TRAP2 mice that I generated when I was a postdoc in Leach and Lowe's lab. So the way this works is you have one allele where the FOS promoter is driving expression of both FOS itself and tamoxifen-inducible Cre. And then we can introduce a second Cre reporter allele. This can be transgenic or virally delivered. It's a modular system that can allow us to look at the morphology of the cells, manipulate their activity with optogenetics, record their activity. We can basically do anything we want. And then the way this works is when a neuron becomes active, initially this Cre-ER won't go in and do its job, which is to excise this stop codon and allow expression of this effector gene, unless we also deliver tamoxifen at the same time. And so in this way, we can pair tamoxifen injection with the behavior that we're interested in studying. And so only those neurons will express the reporter that we want to use. And so we use this in a lot of experiments I'm going to talk about today. And so the screen kind of looked like this. The fear condition mice, so these are TRAP2 mice crossed to a transgenic Cre reporter, so any TRAP cell is going to be red. They also had age-matched non-shock controls. And at this point, we decided to actually trap all the cells in the brain that were active during this one-day retrieval, because we didn't want to capture the sensory experience of the shock itself. We wanted to capture sort of the early memory processes. And so then Benita used, and Michael worked on this with her a lot, tissue clearing and light sheet microscopy to clear the brains, and then we image them with a light sheet microscope. And Michael has developed some really nice computational tools that allow us to identify all the cells and count them automatically according to the Allen Brain Atlas. And then the idea was to look for hit regions and then go in and do some more in-depth studies to look at their functional contributions to memory. And so these are what these data look like from our whole brain screen. Here, the data is normalized to the non-shock age-matched controls. So for the infants, we were particularly interested in the regions that fall into these upper right quadrants. So each circle is a brain region, and the ones in this upper right quadrant are the regions that had more trap cells or more active cells in the fear condition mice compared to the age-matched non-shock controls. And so we thought this might be a good place to start. So in the infants, you see regions in the hypothalamus and the thalamus, one cortical association area, but not the regions we typically associate with memory consolidation. Those started to pop up in the juvenile. So here you can see things like retrosplenial cortex, anterior cingulate area, temporal association area, anterior thalamic nuclei. That was actually present in both. But these cortical areas have known roles in memory consolidation. And we also saw a lot of the same regions in the adults. And so we decided to focus on try to understand the development of one of these cortical regions that was present in the juvenile and adult, but not in the infant, suggesting that something major is changing in these regions during the time when memories go from forgotten to lasting. And we decided to focus on the retrosplenial cortex because it sort of sits between the hippocampus and the prefrontal cortex. And there's a lot of really beautiful studies, including from Andre when he was in Mark Mayford's lab, about how this region is really important for consolidating memories. And so we know a lot about what it does in the adult brain, and the development of this region is really poorly understood. So here's just sort of a summary of what some of the things we know about how in adults, how the retrosplenial cortex contributes to memory consolidation. We know that the cells active at the time of learning can be reactivated during memory retrieval. We also know that activity during learning is really important for memory consolidation. There's increases in neuronal excitability and synaptic plasticity that happens after learning. And there's also a couple of processes like protein synthesis so there's an initial wave of protein synthesis that occurs at the time of learning, and you need that to establish a memory that lasts at least a day. And then there's a second wave that occurs offline in the subsequent 12 hours that's required to form an even longer lasting memory. And then Andre had a really nice study that showed that if you tag the neurons that are activated during learning and then you reactivate them offline when the animal is asleep, you can actually accelerate the process of systems consolidation. And so this told us that in the retrosplenial cortex, a set of dynamic processes is initiated at the time of learning and continues offline, and all of this is required to form a long-lasting memory. So how do these processes differ in the developing brain? Since we had the trap mice, we started by focusing on these learning-activated population that people have shown to be important. And the first thing Benita wanted to do was ask whether if she enhanced memory of these sort of memory trapped cells in the retrosplenial cortex, can she rescue these lost infant memories? And so to do this, she used chemogenetics. So she used a preconditional dread. She put that in the retrosplenial cortex, gave some time for expression, and then she trapped animals, she fear conditioned them, and then she trapped them during this memory retrieval test one day later. And then what she did was she delivered CNO offline, and right before a 7-day memory test, we're kind of trying to hit the system with a hammer, saying, if you enhance the excitability of these retrosplenial sort of learning and memory cells, can we rescue these lost infant memories? And it turns out the answer was no, you can't do that. However, if you do the same manipulation in juveniles and adults, you can significantly enhance memory. So these guys will freeze a lot more if you activate these cells. And so what this told us was that in this window between P17 and P24, when we tested these guys, the retrosplenial cortex and its circuits are still too immature to show this kind of plasticity that you can see in the older animals. And so then she followed up with a second experiment. She said, okay, it seems like it's quite immature. What if I do this experiment, but I just wait longer, allow development to progress a little bit further? Then can I sort of reawaken these lost infant memories? And so here the experimental design was a little bit different. She used the same chemogenetic approach. She, in this case, trapped them during fear conditioning, but now she waited a little over a month. So these animals were almost adults at this point. And then she tested them by injecting CNO acutely, either in the fear condition context or in an altered context. And then what she found was that the GFP controls had completely forgotten this fearful association, as we expected. But in the HM3D group, she could bring back this lost infant memory, especially in the original conditioning context. But in an altered context, the effect was much weaker. So this told us a few things. It told us that these retrosplenial neurons that are active at the time of learning can store this latent infant memory. And you can access this memory again under the right conditions. So in this case, she needed to increase the excitability of those cells using HM3D. And also the salient contextual cues also needed to be present. So there seems to be some synergy between the activity of those trapped neurons and the sensory cues coming from the environment. And so I'll just summarize sort of what these initial findings. So what I've showed you is that infants, juveniles, and adults can all form a fear memory and recall it after a day. But after a week, those that learned as infants no longer recall that memory. And at that point in time, you can't even bring that memory back artificially using chemogenetics. However, if we wait longer and use that same chemogenetic approach, you can get this sort of what we call artificial recall, where you can force that memory to come back under the right conditions. So we're interested in both what's happening across development that contributes to this switch between infants and juveniles, and then also what's happening over time in those animals that learned as infants. What's changing that sort of unmasks this latent memory and makes it accessible under the right conditions. So Benita did, I'll show you 2 more experiments. So I had told you a few slides ago that we know that the retrosplenial neurons that are active at the time of memory encoding can be reactivated when you recall a memory. So we wondered if this sort of like ensemble stability may be contributing in some way to the forgetting. And so here Benita trapped neurons in the retrosplenial cortex during fear conditioning. So they would turn, she's using these AI14 mice, so they'll all turn red, and then waited a week, put the animals back in that original context to test their memory, and then stained for FOS to look at the overlap. So if a neuron is active at the time of learning, to what extent are they reactivated when you recall the memory, and how does that change across development? So these are what these data look like. The pink neurons are the trapped cells, the green neurons are the FOS positive cells. And so she quantified the overlap between the two. And you can see there's a significant increase in this overlap between the infant and juvenile stages. And that's maintained through adulthood. And then non-shocked controls just had generally lower levels of overlap, showing that this is learning specific. And so this suggests that perhaps the instability of the memory engram in the infants may contribute, in the retrosplenial engram in infants may contribute to their forgetting. And then we wanted to look a little bit deeper and try to understand whether we might see some sort of synaptic basis for these changes. And so in this final experiment for this first part, Benita then took the trapped 2 mice, and Abby and Brian worked a lot on this as well, and crossed them to these morph3 reporters, which basically allow detailed anatomical analysis of a specific population of interest, in this case, the trapped cells. And so they fear conditioned these animals and trapped them, and then waited a week and counted up all the dendritic spines. And so these are the sites of excitatory synaptic contact. And this will sort of just give us an idea of how many connections these trapped cells have formed depending on age. And so here's an example of what these data look like. Sorry, it's a little blurry on here. But what you can appreciate right off the bat is that infants, whether they're fear conditioned or not, have a lot more spines than their older counterparts. And so we've quantified this data here. So if you just look at the gray bars, these are the non-shocked mice. So this is sort of just looking at what happens during development in the retrosplenial cortex. And you can see there's a lot of pruning happening between the infant and the juvenile stages. And then maybe it continues a little bit more in the adults. And then in the fear conditioned mice, we actually saw a small decrease in the number of spines in the infants, which sort of goes into an idea I'll talk about, that like actually sparser connections are important for maintaining a memory. And then we also interestingly saw structural plasticity. So in adults, they grow more spines after learning. So the fear conditioned mice have more spines than the non-shock controls. We're not 100% sure if this has anything to do with long-term memory, because we don't see this in the juveniles, which also have long-term memory. So it may be that the 2 age groups solve the same problem in different ways. And then we did one more experiment where we wanted to know, so we had seen that if you wait a month, you can reawaken this lost infant memory with chemogenetics. So we wanted to see what the spines look like on those trapped cells that we trapped in infants and then wait a month. And we saw that there's a significant sparsening of the spines at that point. And so I'll kind of put together, we're still kind of wrapping this up, but this seems like sort of sparsity around the time of encoding or shortly thereafter is really important. And maybe also connection specificity may be really important for forming a memory that's going to last a long time. So we've put together sort of a little cartoon model that we are still sort of refining, but this is how we're thinking about this at this point. So in an infant that undergoes this associative learning, our whole brain trap data showed that you get a lot of cells active, but the non-shock controls also had a lot of cells active. So we think that maybe there's some sort of non-specificity at this point. You get a lot of active cells. Some of them may be important for that learning experience, but a lot of them probably aren't. A week later, and also there's a lot of connection. There's a lot of synaptic connection. So a week later, when you present the animal with contextual cues, you don't get natural recall because maybe you're not even reactivating those same cells, because there's probably, these synapses are dense and they're probably pretty dynamic because we know a lot of them are going to go away. So we think this sort of lack of specificity leads to a reactivation of basically the wrong cells. And so you can't couple that experience to any sort of behavioral expression of the memory. And because of this, you also can't get the artificial recall when you have this sort of densely interconnected network. However, if we wait longer and pruning occurs, you lose some of these connections. And at this point, contextual cues still won't drive natural recall. But if you excite those original cells, some of which are probably important for memory, you can access this latent memory. So just to summarize, I showed you that retrosplenial engram cells encode latent infant memories. Non-specific engrams and combined with dense synaptic connectivity leads to a failure to reactivate those cells originally involved in learning and forgetting. And then synaptic pruning over development can unmask these latent fear memories. So this is sort of how we think about this at this point. I think there's a lot of exciting directions to go here. I'm going to go to the second part, but I can maybe stop if there's any questions on this part first, or just continue till the end. Okay.
What may be a naive question. I was intrigued by what you mentioned in the introduction about like reactivating groups of neurons during sleep, being able to be involved in sort of accelerating the consolidation.
Yeah.
And this may not mesh with the data that you have, but can there potentially be a role for differences in sleep patterns?
For sure.
Just the
Yeah.
that reactivation is or is not happening.
Yes, I think, yeah, definitely. And that's something we've thought about a lot, but it's a can of worms we just haven't opened yet because we don't have the expertise in my lab. But I think, but yeah, infant sleep is really different than adult sleep. And so I'm sure it's playing an important role. And I'll have to find I want to find someone who can help us look at that. Yeah. Yeah.
Emerging myelination.
Mm-hmm.
So like,
yeah.
I mean, I'm wondering about, it's like your data is showing that there's like the necessary connections for the latent memories, like they exist.
Yeah.
There's something also about just like emerging maturity of
myelination.
Yeah,
that's a really interesting question. I think,
and that may be more relevant for connections between distant brain regions.
Right, exactly. Yeah, so I don't think actually, I mean, Abby's doing these experiments now. So Abby's like mapping the long range inputs and outputs. And that would be cool to like maybe combine some of this with a myelin stain because we'd want to know if those same neurons. But yeah, so Abby's been doing a lot of chemogenetic inhibition experiments, and it's actually harder than you'd think. Like just inhibiting activity of those cells doesn't interrupt the memory very much. So I think everything is encoded in like synaptic connections, and things like that are going to be way more important than just their overall activity level. Yeah.
I'm very interested in what you said about versatile cells, from solving the memory retrieval problem by different strategies almost.
Yeah.
Do you think if you were to stimulate optogenetically those cells at different stage of development, you would see different reactions to the brain, like a whole brain CFOS staining, for example, like downstream?
Yes, I do, because at P24, it's really like the cortical inhibition is maturing a lot, and so that is something that really shapes long range interactions. So that's a cool experiment, and I do think that like Abby's been looking at like PV cells, and their like perineural nets are changing a lot across this time. But then the goal is to kind of look at how microcircuit changes are changing the way retrosplenic cortex interacts with its inputs and outputs. Yeah. Yeah.
The outsider of the memory, so there's person, I'm just always curious. So why all this memory paradigm, it needs to be their
Yeah.
connection
Yeah.
from maybe
Yes.
how that can happen. It should go in like mouse study.
Yeah, it's a lot harder to study positive memories at the very young age because like they're still getting all of the milk from their mom. Usually it requires some sort of like food or liquid deprivation in order to get them to like seek a reward. And so for that reason, it's been a lot more challenging. But I get this question a lot. There are studies that show like neutral memories also can show this. Like if you do like a radial arm maze where the animal has to remember where something was in space, there'll also be infantile amnesia for that. So that's something that maybe is a little bit more tractable to look at. And probably that one would probably retrosplenial would be a major candidate. But I think, so yeah, and retrosplenial may be important for a lot of the different ones. It's sort of like the first way station after the hippocampus. And it may be that after that point is where you start to see more segregation in terms of like the content of the memory. Yeah. Yeah.
I think that the contextual peer conditioning experience changes the synaptic pruning in some way. Does it influence that formation of an ensemble, so you have that engram that persists?
You mean in the young ones?
Yeah.
No, because we've actually, well, I don't know, but at this level, so now we have data, I don't have it, I didn't put it in yet, but the non-shocked ones look exactly the same. They prune to the same level. Yeah, I don't know. That's something, yeah. These are all the things we're going to start working on now is try to understand which connections are being lost over time. And so, yeah, that would be really cool if it was actually different. So like Abby's been mapping the inputs to the trapped cells so that we can start to, and also like staining for FOS in them. So we can see not only which connections are changing over time, but like which ones are more or less active depending on how old the animal was. So I think once she's finished collecting all data, we'll have a lot of insight into that. Yeah. Okay, so now I'm going to go to the second part, and we're going to move over to the MPFC. So the medial prefrontal cortex is a major interest in my lab. It's required for both recent and remote memory retrieval. And so in this project, we're trying to understand what's the nature of this cortical network that forms as memories transition from recent to remote, which cells are important, which connections are important. And because the MPFC is involved in recent and remote memory, and when I say recent in mice, I'm talking about at 1 day later, and remote is usually like a month later. And we also know that the MPFC receives inputs from the hippocampus and is densely connected to a lot of other cortical areas. And so it actually may be important in driving the formation of this long-term memory network. And so when I was a postdoc, I was working on this problem. This was sort of like the first application of the TRAP2 mouse. And just to summarize what I found was that in MPFC, there's a lot of dynamic turnover. So there's a set of neurons that are active in the prefrontal cortex at the time of learning, but through some process, new neurons seem to get recruited to a memory trace over time. So I told you before that in the retrosplenial cortex, there's more stability in the adults, but it's no means fixed. There's still a lot of dynamic changes happening during memory across time. And I also used several different whole brain mapping approaches and showed that the neurons involved in recent memory have stronger functional connectivity with subcortical areas like thalamic areas and the striatum, whereas those that are involved in remote memory have more functional connections with a set of cortical areas. So, this seemed like an interesting way in which you could form this cortical network, but by recruiting new classes of cells to a memory trace that project to these cortical areas. So when Zach joined my lab as a postdoc, he wanted to test that idea, to ask, are specific classes of MPFC projection neurons recruited to a memory trace that can sort of drive the formation of a cortical network? I know I'm at UCLA, but he's on the job market. So tell your friends if you at other schools if you were excited about what I'm about to tell you. So he decided to focus on the connection between MPFC to a region called the temporal association area, because we actually don't know that much about TEA, but what we do know is that there's a few studies that show it plays a key role in remote memory. So there's several studies that use lesions to show that TEA is important to recall a remote but not a recent memory. And then Michael in my lab and worked with another student, Cassie Clune, to do really detailed mapping of the projections of MPFC neurons and show that TEA is a major target of intratelencephalic MPFC neurons, or cortical intratelencephalic, or MPFC neurons. So we thought this would be a good candidate to start with. And so Zach started by using miniscopes to record the activity of MPFC TEA neurons during recent and remote memory to see if their functions change across time. And so this is what he did. He habituated the animals, fear conditioned them, and then recorded them during recent memory recall 1 day later or remote memory recall 28 days later. And he used a viral strategy to label either just the neurons that project to TEA with GCAMP. And he also had a second set of animals as a comparison. He just recorded from the general population of MPFC neurons, which would include these and many other cells. And then he first looked at the activity changes within individual neurons using RLC analysis. And what he found is that if you look at the MPFC TEA neurons, you see a big gain in encoding of this conditioned tone at the 28-day time point. But he didn't see this to the same extent in the general population. He also looked at how many neurons encoding freezing behavior itself, and he didn't see any time-dependent changes in that. And so this suggested that these neurons are particularly important for encoding the conditioned tone, and also that while you don't see basically a generally stable number of MPFC neurons encode this tone across time, but by the remote time point there's an enrichment of this population, so that more of the population is made up of these MPFC TEA projection neurons at the remote time point. Then he wanted to look at their population activity to try to understand a little bit more of the behavioral relevance of the activity in these neurons. So prior studies have shown that MPFC population activity encodes sort of high dimensional representations of behavior. So they can tell you about behavior in different contexts. For instance, when the tone is playing and threat is imminent versus the intertone intervals when there's not an imminent threat, and they can encode different aspects of behavior in that. So Zach looked at how the population activity encodes the intersection of cues and behaviors and how this encoding evolves across time. And so to do this, he took all the neurons that he had recorded, either in MPFC TEA population, or I'll show you in a second the overall population, and he used an approach called CBRA, which basically uses a machine learning approach to create a low-dimensional representation of the population activity. And that's what's represented in these plots. So each point is a video frame, and this represents the neural activity sort of in this state space, either during the tone, which is represented in purple, when the tone is on and the animal's freezing in yellow, or when the tone is off and the animal's freezing, so we call that contextual freezing, that's in teal here, or just the rest of the time, in which is the animal's just kind of wandering around this box. And so he also calculated the centroids of each of these little clouds, and wanted to know how distinctly is the population activity representing these different behavioral events. And so what he could see was that the MPFC TEA population had an emergent distinction between tone freezing and not freezing. So meaning this activity was really good at telling the difference between when the tone was playing and the animal was exhibiting this fearful behavior versus when it wasn't. So then he looked at the general population, And he didn't see that change. He also saw this distinction between tone freezing and context freezing. So essentially, these MPFC TEA neurons across time are getting really good at encoding sort of different aspects of this memory-guided behavior, suggesting they play a more important role in driving the behavioral expression of the memory at the remote time point. And so next he wanted to test their functional role using optogenetics. And so he put inhibitory opsin JAWs in the MPFC in trap mice, and these are not in trap mice, in the MPFC of wild type mice, and then implanted fibers to inhibit their terminals in the TEA. And he fear conditioned them, and in one group he inhibited the memory 1 day later, and in another group he inhibited the pathway during recent memory, and in a separate group he inhibited the pathway during remote memory so that all of them could have the exact same amount of time of viral expression time. And what he found was that there was no effect in this recent recall group, but the animals were inhibited this pathway during remote recall froze significantly less. And there was no effect on contextual freezing at that point. And so this suggests that this MPFC to TEA pathway plays a selective role in remote memory. But while these effects were specific to the tone, which lines up with what he observed with miniscopes, the effect was relatively modest. And so we wanted to look at where else those neurons project, which would suggest that maybe these neurons themselves are really important, but it's not just this one projection of those neurons that's critical. Maybe they're coordinating the activity of multiple regions during their role in remote memory retrieval. So to look at that, Zach used viral tracing combined with tissue clearing and light sheet microscopy to map the brain-wide axonal projections of these MPFC TEA neurons. And he wanted to look specifically at the ones that were involved in recent memory or remote memory. So he actually has 3 groups here. So in one case, he virally labeled all MPFC TEA neurons. In another group, he just labeled those MPFC TEA projection neurons that are active during recent memory. And then the third group was those MPFC TEA neurons that were active during remote memory. And he did this by combining TRAP2 with a series of intersectional viral tools. And with this, he could generate these whole brain maps of where else those neurons project. So we know that MPFC neurons usually target like 5 or 10 regions at a time. An individual cell will send axons to about 5 or 10 regions. And so even within this MPFC TEA neurons, those individual cells can also target another subset of regions, but we didn't know which ones were the ones that were involved in memory. And so these maps are just showing you, are basically density plots of where else those neurons project. So you can see that when we mapped all MPFC TEA neurons, of course, there's a lot of axons in TEA. They also project to parietal area, retrosplenial cortex, BLA, nucleus accumbens. And then when he looked at the recent or remote memory subsets, they looked pretty similar, but he did find a couple key differences. And I'll point your attention right here to the claustrum. You can see there's a lot darker, that region is a lot darker on these maps. And so when he quantified this, these are showing the behaviorally activated cells compared to the general population. So in both the recent and remote memory populations, they had significantly more axon projections in the claustrum and significantly fewer in the central amygdala. So this suggested to us that these MPFC TEA neurons that are involved in memory represent a specific subset of the whole population of MPFC TEA neurons, but we didn't see any differences between the recent and remote time point. So we don't think that a new subtype is getting recruited at remote memory. Instead, there's some functional change going on. And also that now we know where else these cells project, and so we think that they may be coordinating memory retrieval through their projections to some of these other key regions, particularly the claustrum, and Zach's testing that now. Okay, and so in the final experiment I'll talk about, Zach wanted to learn a little bit more about how these TEA projecting neurons get functionally recruited to a memory trace over time. And so there's been a lot of nice studies that have shown that activity in MPFC during learning is important for memory consolidation, similar to what I told you about the retrosplenial cortex. And we know that there's some functional maturation that happens of the learning activated cells in MPFC. So they grow more spines over time. They also form connections with other neighboring cells after learning and during the subsequent consolidation period. So what the hypothesis Zach was testing was that the MPFC neurons that are active at the time of learning play a key role in recruiting, in functionally recruiting these MPFC TEA projectors to a memory trace across time. So to test this, he used something called tetanus toxin, which you can deliver it to specific cell populations. In this case, he trapped the MPFC neurons that were involved in learning. And then tetanus toxin blocks synaptic release and by blocking vesicle fusion in those cells. And so he's silencing the synaptic output of the trapped cells in this case. So he fear conditioned the mice and trapped them. So all of these cells that are active during learning are going to express tetanus toxin. First, he looked at what happens to behavior. On the next slide, I'll talk about what happens to the PFC TEA projection neurons. But first, he wanted to look at behavior. So at day 1, we don't think there's very much tetanus toxin expressed yet because of the way this mouse line works. So this is sort of a control just to show that both the GFP controls and the tetanus toxin expressing mice learned and formed a recent memory. But by 28 days, there's very little freezing behavior in the tetanus toxin mice. So first of all, inhibiting the synaptic output of these cells blocks memory consolidation. He also did a control where we wanted to make sure the effects were specific to the learning activated cells. And so in a control, he trapped the MPFC neurons when the animals explored a novel environment the day before, and which also activates a lot of cells in the brain. Then he fear conditioned them and did the same experiment. And in that case, the remote memory was still intact. So it's the neurons that are active at the time of learning have a specific role in memory consolidation. And then he wanted to ask if this impairs the functional recruitment of these MPFC TEA neurons. So in the same mice, he had also labeled the TEA projecting neurons with a virus. And so in these pictures, they're yellow. And then after the 28-day recall, he stained for CFOS to see how many of those MPFC TEA neurons were active during remote recall and whether that number was affected by putting tetanus toxin in the fear conditioning active cells. And what he found was that there's significantly fewer FOS-positive MPFC TEA projection neurons in these animals that had tetanus toxin compared to GFP and novel environment controls. So that suggests that the neurons active during learning initiate a dynamic process that's important for the functional recruitment of these cells to the remote memory trace. So I can just summarize this quickly. Zach found that these MPFC TEA neurons increasingly encode the conditioned tone across time. He showed their population activity has enhanced encoding of, and more specific encoding of tone-evoked freezing during remote memory. He showed that this MPFC TEA pathway is required for remote but not recent memory retrieval, and that the collateral projections of these neurons that are involved in fear memory preferentially target the claustrum. And finally, he showed that behavioral expression of a remote memory and recruitment of these PLTEA neurons requires the synaptic activity of those PL neurons that are active at the time of learning. So we've kind of put together this little model of what we think is going on. We think there's in MPFC, I forgot to say, PL is just a subregion of MPFC. I was using those interchangeably, but there's a set of neurons that are active at the time of learning. These red cells represent the cells that project to TEA. Over time, there's some offline process that's sort of maybe maturing the connections between those cells so that over time, you instead, when you recall a memory, you're preferentially now activating these MPFC TEA projection neurons. And we think this may be an important way in which you form these cortical-cortical networks by recruiting these cortical-cortical neurons to a memory trace over time. And with that, I'd like to thank everyone in the lab, especially the people that work on what I told you about today and all our funding. And I'm happy to take other questions too.